the faculty excellence and achievements in teaching and in discovery, research in the College of Liberal Arts and Sciences. We do this, of course, to stimulate eager and healthy discussion among our faculty, staff, and students, and the greater Iowa State community. So we're delighted to have all of you here tonight to listen to one of our very best young faculty members. I want to thank the Iowa State University Lectures Program for their role in helping us sponsor this event. As always, they do a very wonderful job. Tonight's lecturer is Professor Amy Andreotti. She joins a distinguished list of LAS faculty members who have been part of this series, which was inaugurated in 2006. Over the past several years, we've had an absolutely fascinating group of researchers and scholars who have shared their knowledge on a wide assortment of stimulating topics. These topics have ranged from talking about the physics of the baseball curve, to woody bamboo plants, to West African primates, to dead poets, to old world Mediterranean antiques, to tomorrow's futuristic nanotechnologies, and last fall, we're all enchanted with a fascinating analysis of Russian humor. The depth and breadth of our speakers is indeed astonishing, and it mirrors the academic diversity found in our College of Liberal Arts and Sciences. We are indeed the heart and the soul of Iowa State University. Tonight's lecture will be as equally informative and every bit as enlightening as our previous presentations have been. Amy Andreotti will speak to us tonight about how our bodies fight infection, the basics of molecular immunology. This is a relevant topic to all of us who have been sick at some point. Amy is a professor in the Department of Biochemistry, Biophysics, and Molecular Biology. She researches how specific molecules work, particularly how they signal that there is a viral infection in the body. Tonight she will give us a visual picture of how the body's cells and molecules work together to fight infection. She will also show how damaged or disabled molecules can lead to immune problems. Dr. Andreotti's research could lead to a better treatment of infections and diseases. Her research is also important as some viruses have adapted to the immune system and now can invade the immune system response. Amy Andreotti earned her PhD in chemistry from Princeton University and completed a postdoctoral fellowship at Harvard University. She joined the Iowa State faculty in 1997 and recently received an ISU Award for Mid-Career Achievement in Research. Ladies and gentlemen, please join me in welcoming the College of Liberal Arts and Sciences Pre-Spring 2010 Dean's Lecture Series speaker, Dr. Amy Andreotti.
Thank you. Thank you very much for that nice introduction, and thank you, Dean Whiteford, for giving me the opportunity to speak here tonight. I'm looking forward to sharing with you some insights into your immune system that perhaps you haven't really thought about before, and I think you'll hopefully take away a more detailed understanding of how it all works, although it is a rather complex system, and so we're only going to really scratch the surface tonight. So I'm going to talk about immunity in a way that your doctor does not talk to you about it when you're in his office and you have a sore throat and you're talking about these symptoms and getting this prescription. We're going to go deeper into what your immune system is doing at the cellular and molecular level. And my laboratory is studying how a healthy immune system works. And the reason for this is that it is complex, and we want to understand precisely what the mechanisms are that control it, because when the immune system does make mistakes, it can lead to a variety of very serious conditions and illnesses. And so by understanding how the healthy immune system works at a very detailed level, we're able to perhaps have a better understanding of how to fix it when it goes awry. So this will be the last sort of picture outside the body that we're going to see, and we're going to sort of go in now into the cells and molecules that make up all of our bodies. So this is a schematic of a human cell. You don't need to pay attention to all the labels here, but just to give you a sense of the structure of a cell, there's this outer membrane that encases all of the contents inside the cell. The cell nucleus, which you're probably all familiar with, sort of the control center, telling the cell how to respond to various stimuli. And in all of these things are different substructures, organelles within the cell that carry out various different jobs in the life cycle of this cell. And in here are all of these very important molecules, which we're going to spend some time tonight looking into, mostly protein molecules, which are really doing all the work that's going on in all the cells in your body. You can see the number of cells in your body is quite high. I have to say this, when I first realized how high this was, it made me start to think of the deficit. We're not quite there, but maybe. So we really walk around every day with 100 trillion cells in our body. And they're all very specialized, and you know that. You know your skin cells are different than your muscle cells, and so on and so forth. So the immune system has its own set of specialized cells, and they are collectively called the white blood cells. This is a picture of your blood. These are red blood cells, and in among the red blood cells, which might look familiar, are these white blood cells. And this collection or family of cells is really responsible for mediating your immune response in the case of infections. And there's many, many different types of white blood cells. And this just gives you a sense of all the different cells that specialize in fighting off infections. I'm not going to talk in a lot of great detail about any one of these. You can see by some of the names here that they sort of tell you what these particular white blood cells are doing. These are killer cells and helper cells, and others have less clear names. What I'm going to talk to you about today is mostly, I'm going to just talk about immune cells. And for those of you in the audience who know a little bit about immunology, I'm really going to be focusing on these two types, T cells and B cells. But I'm going to talk generically about immune cells and how they work to respond to an infection. So I mentioned that my laboratory is really interested in the molecules that are really doing the work of the cell and really responsible for all of the cellular function that's going on in your body every day, including your immune response. So on this slide up here are some representations of some protein molecules. We're going to talk more carefully about what proteins are. Unlike cells, which you can see through a microscope, these are way too small to be able to see. And so these are representations that we use to indicate what these proteins might look like. This is showing us what the surface of this molecule looks like. This is called a ribbon diagram. This is an all-atom representation of a protein molecule. And we'll talk more about proteins themselves in a few minutes. These are some nucleic acids. This is a piece of DNA, again, a representation. Bound to a protein molecule, and this is a piece of RNA. And all of these molecules are filling our cells, carrying out very specialized functions. This is a picture from a recent review article that I rather like because I think it illustrates the point that people are really starting to believe, and that is that it's this inside of the cell is a very crowded environment. These molecules are packed very closely to one another, influencing each other, perturbing each other's environment, and in that way carrying out their function. And so I think this illustration where this is the cell membrane and the purple, blue, and orange and yellow are representing different protein molecules as they might be inside the cell. So I think scale is a very important thing to consider before we go much further in talking about these cells and these molecules. And so I'm going to switch over to something here to give you a sense of just how small these molecules are. So I'm going to be zooming in here, and we're starting with everyday objects, a coffee bean, a grain of rice. Here's a sesame seed. And if you care to look, there are unit measurements here. These are sort of the dimensions of these objects. And so as we zoom in, let's see, you can see how things, okay, here we go. You ready? So there goes the coffee bean and the rice. There's a grain of salt. We get into some paramecium here. The human egg cell is, as you know, one of the largest cell. Here's a skin cell. You can see these are things we can visualize under a light microscope. There's the sperm, the red blood cell that we saw a picture of a moment ago. Here's an X chromosome. And these are some of these organelles that I pointed out in the first picture of the cell. So these are the size of those organelles that are inside cells of approximately that size. Next to that is actually a bacterium. And as we move in here, we're moving into the world of viruses. They're significantly smaller than bacteria. So past the viruses, we're finally going to get to some protein molecules. This hemoglobin, I will show you another picture of that protein later. So that's a protein. This is an antibody. This is a sugar, a single sugar molecule. And this is water and a carbon atom right here. So I think that gives you a sense. So now I think it's actually helpful to leave these and move backwards. So we are going to be talking a lot about proteins tonight, and you can see how small that is as we... Oops, let me do that again. Okay. Zooming out past the viruses. Viruses are significantly smaller than the cells they infect. And we're back to the coffee. Okay, so even though I'm going to be showing you pictures of proteins here tonight that are huge on this projection screen, they're really very, very small, and we can't study them directly, and I'll talk a little bit about how we do study them. Okay. Sorry, I've got to find my way around here. Okay, so back to the immune response. I have another diagram of a cell here, and I want to talk to you a little bit about some of the machinery that's at work that helps our immune system recognize viruses when they're present in our bodies. So this is, again, a human cell, and there is this machine called the proteasome, and it's essentially the garbage disposal of the cell. Whenever proteins are, need to be degraded or, put in the trash, they get funneled into this structure. It's a little machine, and that basically breaks this proteins up into small pieces. This is happening all the time in your cells, and the small pieces come out the other end and then get loaded into a protein complex that gets presented on the outside of the cell. And this is called the MHC complex. Sometimes biology is a big alphabet soup, and I'm going to very much try to avoid using that, but MHC I'm going to talk about tonight. So I will be talking, you'll be hearing that term a lot. It stands for Major Histocompatibility Complex, which it doesn't matter what that stands for, but it is the protein that basically is holding little pieces of your cellular proteins out on the surface of your cells all the time. And this process is what is going to signal to your immune system that you have a viral infection, because when a virus enters the cell, and here's a structure of a virus, this is really much smaller than this cell, as we just saw. The virus is made up of a coat of proteins that surrounds the genetic material of the virus, and those coat proteins get treated like basically processed through this garbage disposal, just like your cellular proteins do. They get loaded into this MHC complex and presented on the outside of the cell. And your immune system is able to detect when there's a viral fragment being held out there on the outside of the cell and when it's just one of your normal cellular proteins. And so when it's a viral fragment, that's like a red flag to your immune system. Something needs to be done about this. So this process allows each cell is constantly holding out little pieces of protein, and when those pieces come from a virus, your immune system is able to detect that. And we're going to look at that in some detail. So let's now look at immune cells. How do they detect this? Well, your immune cells have receptors or antennae that they also have on the outside of their surface, basically carrying out surveillance of their environment. And so here's the white blood cell, and this is indicating this cartoon as the receptor. And it turns out that all of your white blood cells, all of the immune cells in your body, have distinct and different receptors. And this is what permits your immune system to be able to recognize really any pathogen that you're going to encounter in your lifetime. It comes from the diversity in these different receptor molecules. So putting these two things together, we have our immune cells with the receptor that are basically going around and looking at these MHC complexes on these presenting cells. And when a connection is made here, that initiates an immune response. So in the next couple slides, I have a few movies to show you because we can see these cells. We're going to look at the cellular level first. We can see how these cells are checking each other out and responding via this interaction here. So this is the immune cell receptor and detecting the MHC complex. So the immune cell in this experiment is in green, and the MHC presenting cell is in red. And this movie comes from the Callahan lab at UC Irvine. And what I'd like you to do is basically pick a pair. And what you're going to see is that the green cell, the immune cell, is constantly following the red cells. It's very unidirectional. It's one way. The green cells are really checking out, is this self or non-self? So you can see, I think, each of these green cells is sort of following the MHC presenting cell around and really trying to establish, is this something I need to worry about? Is there a red flag there, or can it be left alone? Okay. So I have another movie here, and in cell biology, there's many different ways that one can visualize cells. So instead of using the red and the green labels here, now they're using a dye that's indicating the contents of the cell. So this is the immune cell in blue or purple, and this is the MHC presenting cell. And in this movie, what you're going to see, the immune cell is going to turn and identify that there is a viral fragment being presented on this cell, and it's going to change color here, and that's the indication that the immune response is being initiated. So, you want to see it again? Okay. Okay, so it turns, it identifies that this is not a self entity being presented by the MHC, and that color change initiates the immune response. We're going to talk in a little bit about exactly what that immune initiation, what that is. Okay, so the immune response, as far as we're going to go tonight anyway, is leading to cell proliferation and, in some cases, antibody production. So that's something that you're probably familiar with. You know, when you get a virus, your immune system's producing antibodies for that virus. And so all of these cellular processes that we're watching are what initiates that immune cell proliferation. Many, many copies of the immune cell with that particular receptor are made, and then depending on the cell type, they start to pump out antibodies toward that particular piece of the virus. So all of this that we've been looking at is controlled by protein molecules, and this is where my laboratory is really interested in looking at the details of these protein molecules and how they work. And so I'm going to move into that, but first I actually want to show you one more way to visualize this cellular response, and by doing that, I want to emphasize the importance of the proteins that are in your cell. So this experiment, this is an immune cell, and the green here are basically MHC-presenting molecules that have pieces of virus in them. This is not a cell. This is a glass plate. So basically the experimentalist is fooling this immune cell into thinking that this is an MHC-presenting cell, but they're just arrayed on a glass plate. The immune cell goes on, and the camera sits here. And so we can watch this connection that we saw in the last couple movies from the point of view or the vantage point of the MHC-presenting cell. So the immune cell is going to be coming right at us and making its connection to the MHC-presenting cell. In this case, it's just a glass plate. So in the next slide, we can see that movie. So the one on the left is the normal immune cell response. Okay, so you're imagining you're the MHC presenting cell, the immune cell is coming at you. The green here is indicating the location of a particular structural protein in the cell that rearranges to this very regular shape to make a really good synapse or connection with that glass plate, or in reality with the MHC presenting cell. So is that clear what you're seeing there? You can watch that one more time. This is the normal immune cell. So the movie on the right that I'll show you next is the same exact experiment, but one protein has been taken out of that cell, and it happens that the protein that's missing is the protein that my laboratory studies, and this is one of our collaborators at the University of Pennsylvania. So missing one protein, you can see the cell really just can't do it. And this structural protein, which really needs to form this nice clear ring, just can't do it. And so we can watch that one more time. So this is really just to illustrate the importance of even a single protein in this entire cellular process. Okay, so with that introduction to the importance of proteins, let's talk a little bit about what proteins are, where they come from, and what they do. So on the left, I'm sure this is very familiar to many of you in the audience, is the central dogma of molecular biology. This is our DNA, our hereditary material codes for all of the proteins that are made in our body, in all of our cells. So the DNA is transcribed into RNA, and then there's machinery in each of our cells that takes that RNA and makes protein out of it. So what is protein? Protein is basically a polymer of all of the 20 naturally occurring amino acids that are in your body. And I'm sure for those of you who are not biochemists here, you've heard of amino acids when we talk about tryptophan after we eat our turkey, or glutamate and MSG. These are all amino acids. And there's 20 amino acids that vary with respect to their chemical properties. And the protein molecules are basically beads on a string, where each of these beads is a different amino acid that has a different chemical property. And so we can really think of proteins as a bead on a string. So this string doesn't stay floppy and string-like. Protein molecules, in most cases, tend to fold into very compact shapes. So this is just to illustrate this long string. I'm not showing the beads now, just because when we look at protein structures, it's easier to follow the backbone of the protein, or the string part, and that most of the pictures I'm going to show you are just following that backbone. So this is an unstructured, rather random, waving around flexible thing. And these protein polymers fold into very compact or very specific three-dimensional shapes. And you can see in this picture, there's some regions that are helical here, sort of like a barber pole type of pattern, and there's other regions that are, we call these strands or sheets, where the protein polymer is stretched out and elongated and going back and forth like this. And so these regions of regular structure then pack together to make this overall three-dimensional structure of a protein. And the three-dimensional shape that you're seeing here is incredibly important for a protein's function. And I think one of the most striking examples of this is shown on this slide. This is the structure of hemoglobin, and it, you can see, has many of these helical components that are all packed together into this three-dimensional shape. And this is the molecule that carries oxygen in your blood. And I'm sure you're all familiar with sickle cell disease, which is characterized by this sickle-shaped red blood cell. And it turns out that the cause of this disease is a single amino acid change. So one single bead in this protein out of 600 is changed. Let me restart that so we can see the one that's changed. I've colored it in red so you can see when you see it. There it is. So that amino acid right there, in this case, I'm showing mostly all the backbone or the string part, but here I'm showing you the actual amino acid itself. In normal hemoglobin, that's a very polar, water-loving, hydrophilic side chain. It likes to be in the water that's surrounding this protein, because we are made of water. And so all the black here you can think of as aqueous water environment. And in individuals who have the mutation that causes sickle cell anemia, that particular amino acid has been mutated at the genetic level, so the DNA that codes for this protein is changed, and it makes that a very greasy, oily amino acid. So the chemical nature of it's changed. And we all know oil and water don't mix, and so what happens is these hemoglobin molecules clump together in order to try to get that oily, hydrophobic piece out of the water and bury it like this. And so what happens to the blood cells is they take on this rather sickle cell shape because of the change in the overall structure of these hemoglobin molecules as they clump together. So I think this is a really vivid example of how critically important protein structure is to function. Okay, so we are going to get back to the immune response, but I want to spend one slide on how protein structures are determined in the laboratory. And there's two experimental techniques that are used to solve protein structures. The first is nuclear magnetic resonance spectroscopy, or NMR. This technique studies proteins in solution, so they're dissolved in water. And it's actually the same physical technique as is used in magnetic resonance imaging, or MRI, in the hospital setting, which some of you may have experienced. In the hospital setting, they evidently dropped the word nuclear because it made patients nervous, even though this has nothing to do with nuclear energy and everything to do with the behavior of the nuclei in each of the atoms in the protein or the molecule that we're studying. Now, we could talk a great deal about how this works. I'm not going to do that to you tonight. We teach lots of courses in this, so for the students in the audience who want to know more, you can come to class. And this is the NMR facility that we have for biomolecules at Iowa State. The chemistry department also has a few NMR spectrometers. The other technique is X-ray crystallography, or protein crystallography, and in fact most of the protein structures that have been solved are solved using this method. And this involves crystallizing the protein into a solid state, diffracting X-rays off it, and then deducing what the structure looks like based on the diffraction pattern of those X-rays. So in both of these techniques, we are acquiring data on these molecules, so it's a rather indirect way to look at these molecules. We can't see them, and then we generate structural models that are consistent with that data. So all of these protein structures that I'm showing you are models of what these molecules look like in their, presumably in the cell. So unlike looking at cells directly, we have to look at these molecules indirectly. All over the world, there's many labs that use these techniques and solve protein structures. And there's a database called the Protein Data Bank, where once you've solved a structure, you deposit the three-dimensional coordinates of that protein structure into this database. And so it's publicly and freely available. And so if there's a protein you're interested in, you can go to this database and ask, has the structure been solved? And you can download that structure and visualize it on your own computer at home. And I checked this week, at this time, since this was started, there have been 63,000 structures deposited in the Protein Data Bank. I actually looked last week and it was 62,000 something, so it's a rapidly growing database where we're getting more and more structures of these proteins that make up our cells and carry out all these functions. Okay, so back to immunity. You've seen this picture, and what I want to do now that you know something about how to look at protein structures, I'd like to revisit these molecules, these proteins, the MHC complex, as well as the receptor or antenna on the immune cells, and learn a little bit about how these really might look and how they work at the molecular level. So on this slide, this is our cartoon again. This is our MHC presenting cell. This is the MHC protein presenting a little piece of virus that's been passed through that proteasome garbage disposal and presented on the outside of the cell. This is the structure of this protein complex. So here is the outside of my cell. This is my cell membrane, and here's the inside of my cell. And this is being held out into the environment outside of the cell. You can see several features I'd like to point out. One, the viral piece is peeking out here. This darker brown stick is the little piece of virus that's being held at the end of this protein complex. You can see there's lots of these regions. Again, this is really just tracing the string of the backbone of the protein. Lots of these strand regions and two regions with this helical shape up here. This is really the business end of this molecule. You can imagine that where the virus piece is sticking out, that's what we want the immune system to see. And so all of this, most of this protein, is sort of serving as a structural scaffold and holding this thing out there. And so I think it can bring to mind a radio antenna. Most of that is the structure holding that small antenna up into the air. And this is very much the same way. And you'll see this in a lot of proteins. There's a lot that's structural and holding things in just the right position so that they can be, in this case, recognized by your immune system. Okay, so on the next slide, I'm going to take this molecule and I'm going to turn it 90 degrees so that it's facing right toward you. And we're going to then look right directly into the pocket that's holding that piece of virus. And that's shown here. So you can see these two helices now very well. And this is the little piece of virus that's being held out for your immune system to take a look at. Behind here is that large structural part that we really can't see now that we've turned it 90 degrees. This is, I think, one of the most elegant structures in structural biology. To me, it's just so amazing that it's really holding this thing out there like you would hold something out to show another person. But I have to say that maybe the word elegant isn't quite appropriate because it always reminds me of a hot dog, where these are the buns and the hot dog is the little viral fragment that's sitting in there. Okay, so what does the immune cell side of this look like? So here is what we've just looked at. This is the MHC complex. It's being held out by the MHC presenting cell. And this is the immune cell receptor. And we have structures of these two things coming together. And that's what I'm showing you here. This is, in gray, is the antenna or receptor on the immune cell. And again, what you can see is this part down here is really what's feeling the viral fragment that's being presented by the MHC. And most of this protein, again, is the structure or scaffold that's holding it out there. The immune cell surface would be here, and the MHC presenting cell surface would be here. So just to remind you about this movie, when this connection is made and indicated by this color change, really what's going on between these two cells is these proteins are coming together and contacting each other, and you can think of it as your immune system is checking out, really what's sitting in that pocket of that MHC complex. Is it something I know, or is it a virus or some foreign pathogen? Okay, so let's look at that interface a little more carefully. And a lot of structural biology, we look at the details. And I'm not showing you all the details here because, again, I'm just showing you the backbone or the string, not the beads. But it turns out there's 6 fingers or loops that sit down onto the viral presenting MHC right here. And I've colored them hot pink because this is where your immune system gets its ability to recognize any pathogen that you're going to encounter. Our DNA is coded in a way that we get an incredible amount of amino acid diversity in this region. So we can do the math. So I mentioned earlier that all of your immune cells carry a different receptor, and this is where they differ, just in these 6 loops, pretty much. And the differences here allow one of your immune cells, hopefully, when you get an infection, to recognize this particular virus that's being held out there. And then it makes copies of itself. So if we just assume that there's 10 amino acid positions in each one of these loops, that's 60 positions. And so if you do 20 amino acids in 60 positions, I haven't done the math, but it's a big number, 20 to the 60th power. And so your immune system has this incredible range, and you're walking around every day with amazing diversity in this region of these receptor proteins. Okay, so now I think we've pretty carefully looked at what's happening outside of these cells when they're recognizing one another. And so what happens next? We saw that color change. Let's talk about what's happening inside the cell. So the name for what happens next is cell signaling, and this is really where my laboratory is focused on. And it's basically a cellular game of telephone. You've all probably played this game when you were a kid, where you have a long line of children, and at one end they start with a sentence and they whisper it to their neighbor, and it gets whispered down the line. And the fun part of that game is how that message changes as it comes out the other end. And clearly in cellular signaling, you don't want that message to get garbled, or else you're going to have very serious disease problems. So cells must communicate whatever signal's happening on the outside to the inside. And remember, the nucleus down here is the control center and is the part of the cell that's really going to tell that cell how to respond, to proliferate, grow, and divide, or produce certain chemicals to get some other effect. So I have a movie here illustrating cell signaling. This is the outside of the cell membrane. These are some of the organelles we saw earlier. This is the nucleus. So the first thing that you're going to see is receptors. We've looked at those, the immune cell receptors, and this in our case is the MHC complex. That binding initiates a lot of chemical changes just right inside the cell here. And each of these blobs are protein molecules that are responding to these chemical changes and binding or joining this group of a growing complex of proteins. Each protein communicates to the next, until ultimately a signal is passed into the nucleus, and that cell makes many copies of itself, or whatever the response is needed. So the cell signaling is mediated by these protein molecules. So you can imagine that before this receptor engagement, which is occurring outside the cell, occurs, all these protein molecules must be sort of turned off and quiet and not communicating with their neighbor, because if they were, then you would be sending signals to the nucleus in the absence of any external signal. And in fact, this is one source of diseases like cancer, where a cell is growing out of control. Oftentimes what happens is one of these proteins is switched on, maybe a genetic mistake, and it's sending signals into that nucleus to tell that cell to grow and divide when there's no growth hormone or no signal on the outside to tell it to divide. So you get uncontrolled growth. And the structural biology has actually got some good insight into some of these on-off switches. And in the next slide, I want to show you one of these switches. So these proteins really are turned on and off like a light switch. And this particular protein is a signaling protein that results from a chromosomal rearrangement and causes a certain type of leukemia. And we have structures of this type of protein. This is the protein when it's on, and this is when it's off. So this is the same protein. And what I've done here, again, we're just, to help you look at this, we're just looking at all these helices are, this is just the backbone or the string part of the protein folding up. What I've done in these two examples is the blue part, I've put in exactly the same orientation for you. And so what you can see is that this part of the protein is a big arm. In the on state, it's out like this, and when it turns off, it folds in like that. And so it's really a switch. This protein is actually an enzyme, does its chemistry in this configuration, and it's turned off and unable to do any chemistry, unable to send any signals in this configuration. So this is actually was one of the first targets for a clinically used drug that was developed by Novartis earlier about 10 years ago. And this drug is called Gleevec. Some of you may have heard of it. And this was quite successful. This is the chemical structure of that drug for the chemists in the audience. And this in red is this drug molecule binding to or contacting this protein molecule. And one of the reasons that this was a successful anti-cancer drug is that this protein is made only in cancer cells, and it's stuck in this on position, and that's why it's a cancer cell, because this protein's stuck on, it can't turn off, and so it's sending signals to these cells. It's a leukemia, so these are white blood cells to grow and divide. And so they were able to target cancer cells because this drug only bound to this protein, which was only in cancer cells, and so this was quite successful. But also, I think it gives you a sense of the size of a drug molecule compared to a protein. And we're going to talk about another drug before we're done tonight. And I would say nearly all of the medicines that are developed are targeting specific proteins like this. This one got a lot of attention. It was on the cover of Time magazine in 2001 and sort of touted as maybe a magic bullet. And unfortunately, it's a little more, lots of cancers are more complicated than this. They don't have such a clean, this protein is only in cancer cells and not in normal cells. And so it's difficult for pharmaceutical companies to develop drugs that have that sort of specificity. But in this case, they did, and it is being used. Okay, so another picture of cell signaling, and I don't expect you to see any of the details here, but this is just to show you that it's really quite complicated. It's not as simple as that other cartoon. This is an immune cell. This is the MHC presenting cell. In this yellow box is the MHC complex and the immune cell receptor. We've already looked at that structure. Once that binding occurs, we know that signals passed inside, and all of these colored circles are different proteins. All of these arrows are showing the connectivities between these proteins, who's talking to who, and how the signal ultimately gets transferred into the nucleus, where that cell is told, we need to now mount an immune response, proliferate, produce antibody, what have you. So again, this is very much the directions going from here to here for some response. So I've highlighted another region of this. This is really the only slide I'm going to talk about or show you the molecules that my laboratory works on. We're primarily looking at these three molecules in this signaling pathway, and so we do a lot of detailed structural analysis and trying to understand what the on-off switches are, because what you can imagine is that really any one of these proteins or any one of these arrows could be a target for manipulating the immune response, whether you want to turn the immune response up, down, any one of these things. And the more we can understand about the details of how these different steps work, the more success we'll have in being able to manipulate the immune response. Okay, so the last couple slides, I want to show you one more drug that affects your immune response to give you a different sense of drug development here at the end. And that is this molecule that's called FK506. And this molecule, as well as another one, and I'm happy to talk about this more afterwards if anyone would like to, these are natural products. So these two drugs are clinically used and have actually revolutionized the area of organ transplant, because you can imagine that your immune system is a problem if you need a new kidney. You're not going to be able to take somebody else's kidney and live without suppressing your immune system. And prior to these drugs, FK506 and the other one's called cyclosporin, organ transplants just didn't work. People tried, but they didn't work. And so these are natural products that are basically produced by, in this case, it's a bacteria that was identified from a soil sample. The other molecules from a fungus. And this is sort of coming at drug development in the other direction. The Gleevec example I showed you, people were targeting that specific protein because they knew it was a problem. Here, they're looking through, analyzing components in soil samples and finding molecules that are made naturally by bacteria that actually have some benefit to human health. So that's the case here, and this was developed in the 80s. This is the bacteria, this is the chemical structure, and this is a three-dimensional structure of this molecule. So they found that this molecule suppresses the immune response, but they didn't know how it worked. And so a quick discussion about how you figure out how a drug works is illustrated here. So basically you take some tissue, that I think they used calf thymus in this case. They knew it was immunosuppressive. The thymus is an organ that has a lot of immune cells. And basically they're hunting for the protein target of this small molecule. So they put this thymus in the blender, and then the chemists can make a mesh that has this small molecule, FK506, immobilized, and they can basically pour the blended thymus through here and wash it and wash it and wash it, and everything should wash out except for, ideally, the one protein that is the target of FK506. And they did this, and they found one protein that FK506 binds to very, very well, nothing else. And so they named that protein, they weren't very creative, FK506 binding protein, or FKBP. And this is a structure of that protein. As soon as they found this protein, they thought, we've got it. We don't know what this protein is. Clearly, they'd never heard of it before because they got to name it this. But this must be how FK506 suppresses the immune response. And so lots and lots of work was done. The structure was solved. And here I'm showing you a surface structure because I wanted to point out, if you can see this sort of cavity here in the middle, and this is where you're going to see that the drug binds. And many drug-protein interactions occur in these clefts or little valleys that the protein structure creates. As well, this is oftentimes where the protein itself is doing its chemistry or interacting with other things in the cell. And so here is a movie made by Scott, one of my graduate students. I can't make movies like this, showing you exactly how FK506 binds in that little cleft. And it's really kind of buried in there. And so the community thought, we've figured out how this molecule works. We know the protein it binds to. Well, it turns out it wasn't that simple. And I have this is really the last slide, and it's a little complicated, but I'm going to walk you through it because I think you know enough now to follow this. It turns out that, so FK506, which is really, really small, is going into your cells, if you have a prescription, and it finds FKBP, and it binds to it. That was known. And like I said, they didn't know what this protein did, and they thought it must do something in the immune response, but it turns out it doesn't. Instead, what happens is this protein-drug pair together binds to another protein. So here is FKBP and FK506, and it binds to this brown protein. And the name of that brown protein is calcineurin. And this protein is incredibly important in your immune response. In this whole signaling pathway, this is the gatekeeper protein that is responsible for finally passing this signal into the nucleus to tell that cell to proliferate and produce antibodies. And the FK506 FKBP pair basically stops the action of calcineurin. And in that way, it stops the immune response. So it has nothing to do directly with the target of that drug, but rather a consequence of that target drug complex. I think FKBP has really been hijacked by this small molecule and used to stop the immune response by stopping the actions of this protein. So I illustrate these two cases because to really underscore the complexity of these systems and how different drugs work different ways. And we're really far from being able to design drugs, and we really are still learning about the mechanisms that control all these processes. If you are interested in this FK506 story, there's a very good book that is written by Barry Worth called The $1 Billion Molecule, and it's about the development of this immunosuppressive agent. You can see they compare it to a John Grisham novel, so it's really a good read. And it's about early days of organ transplants and the use of these small molecules, and then the race between an academic lab at Harvard and a small biotech startup company to figure out how this molecule works. And I see a couple of people here from my immunology class last fall, and we read this as a class and had fun with it. So I recommend it if you're interested in learning more about this. You don't need to know any immunology to enjoy this book. So with that, I want to just summarize some key points. And I think the key point really to take home is that these protein molecules that are filling all of our cells and doing all of the important jobs within our cells are incredibly intricate. And yet we're able to figure out what their structures are and just how they work, how they're turned on and off. Sometimes we're able to interfere with their actions with small molecules, or in other words, drugs. And that these three-dimensional shapes of proteins are very closely related to function. So hopefully you've gotten a sense of what's happening when next time you're fighting a flu virus. Hopefully not. I didn't get a chance. I didn't really talk specifically about the work that my group does, but they're all sitting right here tonight, and here they are. And it's really a pleasure to work with such wonderful people every day. So here, Raji is a senior scientist in the lab who really is. She's at the bottom here because she holds everybody up, and Stephen and Leah and Scott are graduate students. Sunny is a postdoc. Actually, Leah's a postdoc now. She was a graduate student, and she's leaving in a couple of weeks for her first job. So Shuren is currently rotating the lab, and Jackie's an undergraduate. And David is working here for a year while he puts out medical school applications. So I think, did I get everybody? I think so. So they're really a wonderful group, and it's a pleasure to come in and work with them every day on these systems. So here's some references if you're interested. This one in particular is a nice read. It's a Scientific American article about cell communication and cell signaling, and I'm happy to take any questions.
Thank you very much, Amy. Questions? Are you all feeling healthy? And if you're not, you understand why. So, please. Somebody has to be first.
I'll go first. Amy, you've shown a cell with the MHC on it. About how many MHC molecules would a cell have, and would they all be holding the same thing?
That's a good question. There's a lot, and I really simplified it here. And the same thing with on the immune cell side. There's many, many receptors, and they're quite mobile. They tend to cluster when they start to connect with another cell. Are they all holding the same thing? No, I don't think so. But they're at a moment in time, they're probably all holding something that's related to the same viral infection. And the distinction between self and non-self is gonna hold regardless of whether all the MHCs are holding the same, fragment. So it's a good question.
So your FK506 binding protein, it was hijacked by this drug. Does anybody know what it does normally?
Yeah, it's a good question. So I didn't get into a lot of the details. The other drug is called cyclosporin, and you're probably familiar with that. And it also does the same thing. It suppresses the immune system. It binds another protein, which they named cyclophilin. So you can see these two proteins were discovered because of these drugs and the interest in these drugs. And both cyclophilin, the protein that binds cyclosporin, and FKBP have the same enzymatic activity. And so for the specialists in the audience, they are peptidylprolyl isomerases. So they catalyze the interconversion between cis and trans proline bonds. So and they're very abundant in all of our cells. And so there's some thought that they are protein folding chaperones and that they play some role there. They're probably also regulatory binding proteins. If you look in the literature, there's hundreds of papers on both these, and there's lots of different functions. So I think they do a lot of things, but that is what their enzymatic activity is. And what they do with it, well, actually, cyclophilin is been implicated in HIV infection. There's a prolyl isomerization event on one of the capsid proteins on the HIV virus, and cyclophilin seems to be required for HIV entry into the cell. So in that case, it's being hijacked by the virus. But so I think that they're multitaskers.
Yes, please. And you're next.
Is function of FK506 known? What does it do for the bacteria?
Oh, that's a good question. And I don't know the precise answer to that, but I think a lot of these natural products that we're pulling out of these soil samples are probably defense mechanisms. Yeah. And
Against other bacteria?
exactly, yeah. So they're rather toxic molecules. One of the things I didn't say, but it was the case with cyclosporin, which is also, it's come from a fungus. That comes from, I'm blanking on the name of the company, in Basel, Switzerland. I can't remember the drug company. If anyone knows it, tell me. Anyway, this was back earlier, actually. That was discovered first in the 70s. And at that time, these large pharmaceutical houses would send, when their employees went on vacation, they would send them on vacation with plastic Ziploc bags to bring home soil samples from wherever in the world they were traveling. And that soil sample came from Norway that allowed the discovery of cyclosporin. So it went from, someone went to Norway on vacation, went back to Switzerland with this dirt, and they identified the first immunosuppressive. And if you read that book, it gives a nice history of the area of organ transplantation, and cyclosporin was actually the first drug to permit successful organ transplants. Again, it's probably a defense molecule from that organism.
Yes, please.
What do you think about autoimmune diseases? For example, like Guillain-Barré syndrome, where it's immune response to the, I think it's the central nervous system's outer line.
Mm-hmm.
How the immune system recognizes Helicobacter pylori. When we think that the immune system is regulated by our genetic material, how do you hypothesize that these things develop? Are they environmental? Are they genetic? Is it kind of like an evolutionary process?
That's a good question. And as a structural biologist, we're getting a little away from my field, but I can tell you what I think. So I emphasize self and non-self and the ability of our immune system to discriminate between those two. And you probably know that a lot of what happens during development in the fetal development is that your repertoire of immune cells are being selected for those that don't recognize self during that stage of development, and then those cells die. So ideally, you're born with no immune cells that can recognize your own yourself. And so, I don't know whether autoimmunity, probably if it develops later in life, it's more likely due to, genetic changes that might shift the population of immune cells toward, sequences that can't distinguish between something that's similar to a bacterium. I suppose if you have an autoimmunity earlier in life, there could be something wrong with the system that is preparing you before you're born to not recognize self. So I think there's probably lots of different mechanisms that are going to lead to different autoimmune consequences. And
There is the theory that some, if not many, autoimmune disorders have a parallel to microbial pathogen infection. That's why I was asking the question.
yeah.
If you had ever evaluated any kind of relationship.
Yeah, I personally haven't, but it's probably in a review article somewhere.
Other questions? Yes, please.
About a month ago, there was an announcement that they were studying the Ebola virus, and I was confused about why there was no immune response. You know about this? No immune response.
Well, I don't actually know very much about this, but and
There's no immune
Yeah.
response to the Ebola virus, and they found out the Ebola somehow blocks a protein, which essentially prevents the immune response altogether. Do you know what the protein is? They
So-
didn't elaborate.
unfortunately, my colleague who studies Ebola is not here tonight. I think he's. Where is he? He's traveling somewhere to collect data, I think. So, so I'm not even going to hazard to begin to guess because I really don't know as much as I should about Ebola since he's my next door neighbor in my office. But I would be happy to give you his contact information. I'm sure he would be happy to tell you everything about Ebola. Yeah. I think my knowledge ends at hot zone.
Do you think this discovery is going to promote more discovery of more immune suppressive drugs that would work like Ebola?
It maybe, yeah. I mean, I think I think that immunosuppression is a tricky thing because you don't want to have no immune response. So you have to titrate your immune response in order to not reject that organ, but also be able to fight that cold that you're bound to get. So, we probably don't want to go as far as Ebola has gone in suppressing our immune response. It's a good question, though.
Ladies and gentlemen, we have some beverages and snacks over there, and please join me in thanking our spring 2010 Dean's Special Lecture.
Thank you.